Hi, am I audible? Just a second. Alright, I'm having cold today, so little bit voice is low. Okay, so let me just check once. Okay, so today we are going to discuss about the structure of a seed. Okay, so seed structure we will see. Okay, and what all parts are associated with the seeds we are going to discuss. Okay, and along with that, how exactly seed formation occurs. So what could be the reason? So we will discuss that as well. Okay, so before that, let me introduce myself. I am Vani Kari, biology educator at Unacademy. I am an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. Okay, so join with us in Telegram for homework and class notes. You can join the channel Vani Kari ICSE class 9 and 10 biology. Okay, and the Telegram channel Unacademy ICSE and ISC boards. So this link you will find in the description. So you can click on the link and join the channel for updates regarding the classes. Okay, so we also conduct special classes on Unacademy platform. Okay, which are absolutely free. So learn from your favorite educator for free. Okay, so engage with educator via polls. So we will be conducting poll. Okay, so it is like a practice section for you all. Okay, so clear your doubts in the live special class. So whatever doubts you have, you can clear your doubts in the special class. And now for the top special classes, you can visit the link that is there in the description. Okay, so moving on to Unacademy subscription features. I hope I'm audible. I have a little cold today, so that's the reason I'm unable to speak much. So if you don't understand anything, you can ask me. All right. Alright, so learn live from the comfort of your home. Okay, so unlimited access to all the courses will be there. Top educators of India are going to take the class in one platform, that is an academy platform. Then there is regular doubt clearing session. Okay, so complete syllabus will be covered, mentorship and guidance is given, study material is given. Okay, then practice test, live test series, batch courses. So now if you prefer to learn in English or if you prefer to learn in Hindi, okay, or English also, so it is completely your choice. You will have access to all the batches, so you can go to these batches along with your preference. Okay, depending on your preference. So there is daily practice section, weekly mock test series, live quizzes and daily MCQ subjective test series also you will have. Okay, so now for class 9, so this is for class 9, so you can try with 6 months. Okay, so you can see here as you are going for higher months of subscription, the cost is getting reduced. Okay, so you can anyway try with 6 months, and for 6 months it will be around 10,000. But if you look at this one, it's 12 months, it is somewhere around 15,000 for 12 months. Okay, so with the referral code, that is Vani YT, you can use this referral code. Okay, so with this referral code you will be getting 10% discount on the subscription. Okay, so you will be getting 10% discount. So you can go for any of these months of subscription based on your preference. Okay, so let's see what exactly is the structure of seed. Yes, anybody? What is seed? Like from where the seed formation occurs, what is the procedure that occurs. Okay, so now other than that, what all does the seed contain? Any idea? Any of you? What does this seed contain? Or whether after, so from where exactly seed is coming? It is coming from the flower. Okay, so that is after the fertilization. So now we are going to discuss about the flowering plants which give rise to the seed formation, the fruit formation. Okay, so we will talk about the seed formation today. Okay, then we will also discuss about the fruit formation. Okay, so what exactly is fruit formation, or from where exactly this is coming. Now seed structure, when we mention, okay, so yes, it will have a covering, so that is a seed coat. Okay, so there is always a seed coat present, so that is a covering for a seed. Then cotyledons you can find. What is that cotyledon? So it is basically a nourishment. So now nourishment for the developing embryo in case of dicots. Okay, we will discuss what exactly is this one. And the embryonal axis, so we will see. Okay, so just a second, what shall I explain? Just a second, let me just add few slides. I'll add few slides here so that I can explain it to you. Okay, हाय नीट के बारे में कुछ बताइए. सो यू आर इन क्लास 9, अनु or what is it? Okay, I'm sorry, it's not anu, anime it is written. Okay, all right. So NEET exam is basically for those people. Okay, I think you are in class 9. Okay, so it is for those people who are willing to write their get into medical field. So there are, once you write the NEET exam, so you need to qualify. So there are limited seats will be there, so a lot of members will be participating in that. I mean, they will be writing the NEET exam, and very few medical seats are available. Okay, so you need to be on top to get into any of the government medical schools or medical colleges. So for that you are in class 10. Okay, so concentrate more on now itself. So starting from now itself, you can concentrate more on the concepts, that is basics. So since you are in class 9 and 10, when we mentioned, so it is basically giving you an idea. So basic you are studying. So class 11 and 12 when you get into, so that is when you will study a lot more in detail. Okay, so you'll be studying about this lot more in detail. So you can refer many of the workbooks also that are available. So those workbooks are okay. I cannot say the marks as such. Okay, so it is not like that. So depending on the number of students participating, and there will be a cutoff mark. So depending on number of students participating, and the categories are there. Okay, so every year different cutoff will be there. So you need to have a look at which year you are writing, so that you will understand only then, that is when you are writing it. So cutoff marks will be given, so based on that you will be scoring. Okay, so it's not like you will be getting, so I score about, so let's say 100 marks is there, so you are going to score 50. So that is not like that. So it is depending on the number of students, because all the competitive exams are like that. Okay, so depending on the number of students, and depending on the category, cutoff marks will be there. So based on that, the top list will be created. Okay, so government seat, for government seat only I said. Okay, I'm really sorry, I'm a little cold I have, so I'm thinking a lot and speaking. I hope you got this one. All right, so let's get to this point. So that is the structure of a seed now. Okay, so seed structure, you're welcome, Anime. Okay, so seed structure, when we mention, so it is having a seed code. So let's see here. Okay, so now this one, okay, I'll just erase this part, just a second. All right, so now let's see, this is the seed now. Okay, so this is one cotyledon. Okay, and there is another cotyledon that you can find. So these are cotyledons now. So there is an embryonal axis. Okay, so this is the embryonal axis. So now the seed, there is a coat, okay, so there is a covering for the seed always. So on this side also there is one covering, so there is a covering which is present surrounding this cotyledon. So here also you can find a surrounding, that is seed coat surrounding this cotyledon. Okay, so let's see here. So this one can be the seed coat. Okay, then this can be the cotyledon. All right, so this is a cotyledon. So here is the embryonal axis. So let me just make it in different color so that you can differentiate that. Okay, so this one is now a embryonal axis over here. Okay, so we will just write that. So embryonal axis will be them, and there is a plumule, that is shoot system. So let's draw the plumule now, where you will get, so let's say this is a plumule which will go on germinating. So from the embryonal axis, so there will be a plumule, so this will go on dividing, and it will grow to become a shoot system. Okay, so later on it will start developing a leaf. Okay, so there is a leaf development that you can see. So this is basically a plumule will give rise to a shoot system. Okay, so this is giving rise to a shoot system. Then one more thing is what, so there is a root system also. So now this is a radical. Okay, so this part what you see, so this part is radical which will give rise to a root system. Okay, so there can be tap root, or there can be fibrous root also. So now depending on a condition, anything is possible. It can be either, depending on a type of plant. Okay, so it can either be fibrous root, or it can either be the tap root. Okay, so now this one is a root. Okay, so root system is coming from what, root system is coming from radical. Okay, so it is from the radical. Shoot system is from what, shoot system is from the plumule. So this one was the plumule. So plumule will give rise to a shoot system, and this will give rise to a shoot system. Okay, so now this cotyledon, so this is also a cotyledon now. So these cotyledons in case of dicot, so what is dicot, so you can see here, you can divide the seed equally into two halves. Okay, so you can divide it into two halves, whereas in case of monocot there is nothing like that. So one complete seed is there, so you cannot divide it into dividing a nourishment to developing embryo. So let me just write this. Okay, all right, so let's have a look at this. So now seed structure, so we will see what exactly is seed coat. Okay, so moving on to seed coat, depending on the covering that is there for a seed. Okay, so let's see here. Okay, so I'll just draw it and explain it. So now there is a ovule. So this one, let's say this big thing what I'm drawing here, so this is a ovary. Okay, so this one we will just draw this. So this is now a ovary. Okay, so now this ovary attached to the placenta. So we can show, we will just draw the placenta over here. So this is the placenta. Okay, so attached to the placenta you can find that there is a ovule. Okay, so this can be straight, or the straight one which can be like this, or the ovule can be inverted as well. So let's say somewhat like this. Okay, so now this one what you see here, so this is an inverted ovule that I have drawn. Okay, so this is an inverted ovule having its own integument. Integument is what, so this is now a integument. Okay, so integument is the one which later on, okay, so later becomes seed coat. Okay, so seed coat, it is the one that will become the seed coat. So now here inside you can find the embryo sac. Okay, so here is this embryo sac. Okay, so this end opening you see, right? So this is micropylar end. Okay, and towards the other side it is the chalaza end. Okay, so this one is the chalaza end. So now in this case, as you see here, okay, So now this is a embryo sac. Just a sec. So this is a embryo sac. Alright. So there is a egg cell over here. Okay. So then you can also find a synergid. So on the other side you can find the antipodal cells. Okay. So in the center you will find a polar nuclei. Just a second. Alright, so in the end you will find a polar nuclei that is there. So each of these are having their own nuclei. So here also you can find a nuclei, here also there is a nuclei. So this is now a polar nuclei. Okay. Now this seed coat, what you see, excuse me. Yeah. So now you see the seed coat, right? So this is the outer one. Okay, so this one is the outer one, all right, and this one is inner coat. So there are coatings which we call as testa and tegmen. So later on, after the seed formation occurs, so this is going to become testa and this one is going to become tegmen. Okay, so there is a tegmen. So seed coat which is written over here. Okay, so testa and tegmen. So this is the outer covering. Okay, so this one is inner covering, so tegmen. Okay, so then you can find a cotyledon and a embryonal axis. So here you can find a embryonal axis. Okay, so let's have a look at this one now. Okay, so you can see in this, now this is a dicot seed which is shown. Okay, so there is epicotyle. So I'll tell you here what exactly is epicotyle. Okay, so now this point you see, right? So this is now a embryonal axis here. So let me just show you this. Okay, so between the plumule and here is this cotyledon that is attached. Okay, so between the plumule and between this node, embryonal node, so this point, what is then? So this can be considered under epicotyle region. Okay, so this is the epicotyle region, and now between the root and between the embryonal axis, so this point what you see, so this is basically hypocotyle. Okay, so this one is hypocotyle region. So there is epicotyle region and hypocotyle, so they are basically helping in the embryo formation. Okay, so plumule is giving rise to shoot system, radicle, so this one radicle is giving rise to root system. Okay, so now this is embryonal axis where you can see at the node, so this is a node where these particular cotyledons are attached onto the embryonal axis. So now this one is epicotyle between the plumule and the embryonal node, and between the radical and embryonal node, so it is a hypocotyle region. Okay, so covering is a seed coat that we can find. Alright, so now seed is attached to the stalk by the hilum. So what is that? So here I was saying, okay, so this diagram. So now seed is attached to the stalk. So this is a stalk now. So this one we will just say stalk, which can also be called as a funicle. Okay, and now this particular junction, what you see, so this junction is called as the hilum. Okay, so this one is now the hilum. So there is a seed attachment which we see, so that is with the seed is attached to the stalk by the hilum. As it reaches maturity, so seed will be losing the water. So there is a water loss that we can see. So now at maturity it is dry, that is about 10 to 15% moisture is gone. Okay, so 10 to 15% moisture, once it is gone, so this seed is completely dry. Okay, so now it reaches the seed dormancy, that is due to metabolic inactivity. Okay, so there is a metabolic inactivity that can be seen. Okay, so what exactly happens? The enzymes which are there in seed, okay, so enzymes in seeds will get inactive. Okay, so they'll become inactive, so that we call the seed is a dormant. Okay, so seed dormancy when we mention, or dormant seed when we mention. So enzymes in it are inactive. So they are not metabolizing anything. So based on that, so there are no changes we can see. So now the micropyle is the opening for water and oxygen entering. Okay, so now here again in the same diagram, so you see this opening. Okay, so this is anyway it is the fruit. Okay, so ovary will become fruit later on. So now we consume fruit and we are having only the seed. So now this seed which is actually sown into the soil. Okay, so that is for germination because it has lost its water content also. So once it becomes the seed now, okay, so now you can find an opening there. So this is a micropillar. I'm just I had drawn a ovule like this previously. Now we will just imagine the ovule is like this. Okay, so now this one is a micropillar opening. Okay, so this is a micropillar opening onto the seed. So this is the seed where there is absorption of oxygen. Okay, so water and minerals can also be taken in. So you can see in this itself. Okay, so micropyle is the opening for water and oxygen entry. Okay, so oxygen entry and the water absorption, so all that will occur through the micropylar opening that is there. Okay, so what is taken here? Water is also taken and oxygen is taken. Enzymes inside the seed. So now this is a seed, right? So enzymes inside the seed or enzymes which are there in the seed, so they can get activated. Okay, so once they start to get the enough amount of the temperature. Okay, so there is suitable temperature, or we can say if there is a suitable amount of carbon dioxide is available, water is available, the seed will start to germinate. Okay, so we can see the seed germination. So based on that, so there are the changes that are going to occur, so the plant growth can be seen. Okay, so now moving on to formation of fruit. All right, so cell division in the ovary after fertilization. So this is after the fertilization occurs. So ovary wall which was there earlier. Okay, so this ovary wall which is there, so this is going to become the fruit. Okay, and here you could see that there was a ovule which was attached. Okay, so which is now called as anatropous ovule. Okay, so depending on how it is arranged, so now this is inverted arrangement, so we can call it as an anatropous ovule. So if it is like this, it is orthotopic. Okay, so depending on how the ovule is arranged, so this is after the fertilization. So this whole part which is there attached to the placenta of the fruit, so this will become the seed. Okay, so ovule will become the seed, fruit is basically a ovary is becoming a fruit there. Okay, so cell division in the ovary after fertilization forms the fruit. So there is three layers are there, so that is with respect to fruit wall. So there can be epicarp, mesocarp and endocarp. So wall of the ovary forms the pericarp. So this is the whole thing. Okay, so role of the pericarp is seed protection and dispersal. So now inside this itself, inside the fruit, so you can see in here also. So now this was a fruit earlier. Okay, so now we eat off all this fruit. Okay, so what is remaining? So there is a seed remaining. So this seed can get dry, and when we again sow it into the soil, so this will start germinating. Okay, so now fruit is an enlarged ripened ovary, the ovarian wall forming the fruit wall enclosing the seed. So there is a fruit wall that is enclosing the seed. So fruit protects the seed and helping the seed dispersal. Okay, so that means so once the fruit is all the function of a fruit is done. Okay, so this one will protect the seed. So basically first and the foremost thing is protection to the seed is given by the fruit, and let's say we are eating that fruit. Okay, so after that basically it is given a choice, so where it will become dry. Then once we dry it, so this can be sown into the soil for germination. Okay, or otherwise it will also help in seed destruction. So this is with respect to these type of a pea pod. Okay, so let's say this is a pea pod now. All right, So you can see a arrangement over there in the pea pod. So attached to the placenta, so let's say these are all the placental coverings which you find in the ovule when it was there. Okay, so now you can see there is a formation of seed that has occurred attached to this part. Okay, so this is a pea now. So not all the types of fruit will have this type of ovary. Okay, so now this one outer pod, what you see, so this is the pea pod, right? So pea pod is ovary. Attached to the placenta you see these seeds which are arranged, right? So those seeds are basically the ovules. Okay, so ovules, the fertilized ovule is the one we call as a seed. Okay, so this is a seed now. So this will be covered. So this is only we have one, right, one side written there. So seed is the ripened ovule. One second. Okay. Yes, are you all understanding? Any doubt, Ilya? Alright. So seed is basically a ripened ovule. So it contains embryo. So there is an embryo which develops into new plant. So there is a development of an embryo that we can see. So seed coat protects the embryo from mechanical damage. Alright. So if there is any damage that has occurred, so seed coat is the one which is providing a protection to the embryo. So that is from the mechanical damage. So example here, bean seed is given, peas is given. So all those are being providing a protection. Now grain, what exactly is grain? So this is found in case of maize. So I'll just show you this. So now this is a maize. Okay, so we will just draw the corn, or which we see as a maize. Where is that? Alright, here. So now this one is a maize. Okay, so this is corn, or which we can also write it as a maize. Alright. So now this is a maize seed structure. Alright. So what do you observe in a maize? So when you draw it, so you can just see the arrangement as such. So this would be the arrangement. So then you can see a papery cotyledon there. So this is a papery cotyledon having its own plumule and a radicle. So there is a plumule as well as a radicle. Okay, so now plumule is covered by a protective covering. So this one is a protective covering, so which we call as a coleoptile. So maybe we can just highlight it a little more. Just a second. Alright, so now this is a papery cotyledon that you will find. So there is actually the root system, and there is a shoot system that we can see. So now with respect to this, so there is a covering for this one. So this is a covering that we see. Okay, so here also you can find a covering. So this covering, what you see, covering the plumule. Okay, so plumule covering, when we mention, so plumule covering is basically a coleoptile. Okay, so this is a coleoptile, and here for this one, so this one is a radical. Okay, so for radical, what is the covering present? Anybody? So there is a coleorhiza. Okay, so there is a coleorhiza. So now this one, what you see, so this is a scutellum. Okay, so this is basically can also be mentioned as a papery cotyledon. So this is a papery cotyledon that we can mention. Alright. So now this is basically again separated. So this part is basically the endosperm part. Okay, and the whole covering which is there, so that is the aileron layer. So we will just draw it over here. Okay, so just a second. Alright, so this is completely having a proteinaceous covering, so that can be aileron layer. Okay, so this is the aileron layer. This is aileron. Alright, okay. So this is about the concept. So let's have a look at this. Okay. So now it is the fruit wall is combined and the seed coat is combined. So both of them together we call it as a grain. Okay. So now tegmen of seed develop from. Okay. So testa and tegmen. Any idea what is the answer for this one? Any of you? No idea. So tegmen of seed develops from. So in the beginning I said, so seed coat, okay. So seed coat formation we can see, just a second. Yeah. So seed coat formation we can see, so that is outer and inner. So two of them are there. So outer is testa and inner one is tegmen. So that means inner integument. So this is for a inner side. Okay, so this is the inner and this one is outer integument. Okay, so moving on to the next one. Seed may be defined as ripened ovule, fertilized ovary. Okay, so the gynoecium and the embryo. So let me just say about this. Yes, any idea about this one? So seed may be defined as, so as the definition itself says, so this is basically a ripened ovule. How? Because there is a stigma. Okay, so there is a fertilization that we can see. So now you know this one is stigma, so you can find a stigma there, you can find a style there. Okay, so stigma, style and ovary that you will find. All right, so once this is done, so you know there is the germination of this particular pollen tube. So this is pollen grain. So pollen grain will germinate and it will start forming a tube which will later on enter through the micropylar opening. Okay, so we will just draw two of them here, so that will carry a micropylar. So now inside this, so there is a embryo sac. So here you can find there is a embryo sac. Okay, because you know this over here, we will just show one more structure. All right, so you know attached to this one you can find that there is a ovule which is arranged. Okay, so now this ovule, when we mention, so it is having a small opening through which this tube can enter. So now you know it is first taking an entry into this structure called as the synergid cells of the embryo sac. So now through this tube it will be transferring its male gamete. So one of the male gamete will join with the egg cell. Okay, one of the male gamete will join with the polar nuclei of the other one. So after fertilization, so now this is after fertilization, what will happen? So ripened ovule itself is a seed. Okay, so now seed is a developed ovule, fertilized and developed ovule, then developed ovary and fertilized and developed ovary. Yes, any idea on this? Okay, 1 second. No idea. Alright, so this one is basically fertilized and developed ovule. Okay, so this we have seen the dicot embryo. Okay, so there is a picotile part that I mentioned on embryonal axis. Okay, so this is axis above the level of the cotyledon. Okay, so now hypocotyle when we mention, so this is a part of axis below the level of the cotyledon. So that is between the radicle and you know the embryonal node, what was in. So there the hypocotyle is present. So this terminates in a radicle or a root tip. So root tip covered by the root cap. So there is a root cap covering for a root tip. Alright. So now this is a bean, which is a dicot seed. Now if we are talking about a monocot, what would be the condition? So I explained you about a maize seed, right? So monocot embryo is having a single cotyledon at the end of the embryonic axis. So now you know there is a cotyledon, so same as usual. So there is a epicotyle consisting of the plumule, okay, that is shoot apex and some leaf primordia. So now in this case it is enclosed in a halofoliar structure called as a coleoptile, okay. So there is, this is, let's say this is now a plumule, okay. So plumule is covered by a coleoptile, okay. Membranous sheet will be there, so this is called as a coleoptile. So now axis below the level of cotyledon has the radicle with the root. So let's say this is root now, okay. So root is having a protective covering which we call as a coleorhiza. So there is a coleorhiza that is protecting or providing protection to the root system, okay. So now this is wheat monocot. So this is in case of wheat. So you can see here there is a embryonal axis, so there is a development, okay. So zygote is monocot or dicot? So a good question, but zygote is basically a product after the fertilization, okay. So now depending on which plant, under which plant, so let's say pea plant if I'm saying. So in pea plant the pea flower is there. So pea flower fertilization occurs. Whatever zygote is produced, so that is a digote, okay. So now let's say corn plant is there. You know maize, corn cob you eat, right? So butta. So in that plant if the fertilization is occurring and you know zygote formation is occurring, so that is a monocot one. So depending on a plant, zygote can be either monocot or a dicot. So this is going to form, zygote is just one cell which will later on going to become a seed to form a monocot or a dicot, okay. So it is zygote alone is not monocot or a dicot because this is just a product of the fertilization. Is this clear, Akib? Any doubt? Okay. Alright. So now for testa when we mentioned, so you can see here, okay. So you see this tiny hole here, right? So what is this tiny hole then? So you know through this tiny hole which we call as a micropylar opening, okay. So this is a micropylar opening or a micropyle. So through this structure oxygen is taken inside, I mean through that pore, and you know through this pore or you know there is water also that is absorbed. So how exactly this occurs? So now I'm taking this seed into the soil, okay. So I have given it into the soil. So there is a pore over here, okay. So there is a pore, that is small opening. So now you know this seed is going to germinate. So this is going to give rise to a plant. So how exactly this gives rise to a plant? Because for respiration it requires oxygen. So oxygen is taken through the micropyle. Water is also taken through the micropyle. So seed dormancy will break, okay. So seed dormancy, which was earlier that where the enzymes were inactive, so seed dormancy will break, and by breaking the seed dormancy what is going to happen? So there is basically as a seed dormancy breaks, so this will start to germinate. So you can see there is a plumule, there is a radicle that will be formed. So you can see over here. So this is giving rise to a shoot system. This is giving rise to a root system. Okay. So same thing which I explained with respect to you know the scutellum or you know the cotyledon. So you could see that there is a papery cotyledon. Okay. So there is a papery cotyledon, and you know you can find there is a structure. So this is you know plumule will be there, a radicle will be there. So it is covered by coleoptile, and this is covered by a coleorhiza. So this is the one which is shown here. Okay. So now in this case, so this is the position of the plumule and scutellum. So this is scutellum. Okay. So this one what you see here. So this is scutellum, a papery cotyledon. Okay. And inside you can see a plumule along with a radicle there. Okay. Just a second. Alright. So now you know aileron layer is basically a proteinaceous layer. So this is the endosperm. In case of monocot, the nourishment to the developing embryo. So that is done by the endosperm. In case of dicots, it is the cotyledon. Okay. So in case of dicot, okay. So cotyledon does the nourishment. All right. So who is doing the nourishment? It is the cotyledon. But in case of monocot, it is not the cotyledon, it is actually the endosperm. So this remaining part, what you see, so endosperm is the one that produces or provides some nourishment for the developing embryo. So in angiosperm, what is angiosperm? That is a flowering plant. Okay, so this is a flowering plant. So seed develops after the ovule is fertilized. Seeds are classified into various types based on the developmental factors. Okay. So now based on the presence or absence of residual endosperm, so there can be albuminous or non-albuminous seed. Okay. So if endosperm is still there, okay. So if the endosperm still persists, so that is the albuminous seed. So non-albuminous, when we mention endosperm is not there, okay, so there is no endosperm, but instead endosperm is utilized in formation of cotyledons. Okay, so cotyledons are being formed there. So now based on the number of cotyledons, monocot and dicots are there. Okay, so now if remains of nucellus are present, so that we call as a perisperm. Okay, so this is the seed structure which is shown. So you can see these structures. Okay, so you see this one, right? So you see this opening here. Okay, so you see this opening over here, then you can see a opening over here, you can see a opening here. Okay, so all this is basically a micropylar opening. Okay, so through which the water and the oxygen can be taken inside. All right, so scutellum is papery cotyledon of the monocots. Okay, so now protective layer of a radicle. So radicle, covering the radicle, what protective layer is there? So that is the coleorhiza. All right, so in ex-albuminous seed, the food is stored in case of cotyledon. So that is a non-albuminous seed. So in albuminous seed, the food is stored in the endosperm, and in non-albuminous seed, the food is stored in the cotyledons. Okay, so this is the right answer. So before I end this session, so let me just say about certain, the batches that we have. So we have batches. There is a guardians batch. So this is an ongoing batch. 6th of April it started. So but you have a option to watch the recordings as well. Okay, so you can watch the recordings that are available. Okay, so any doubt you have, you can get in touch with the telegram. Okay, or you can also use ask a doubt feature. So now there is a Eternals batch as well. So start date is 20th April. So this is going to start from 20th of April. So all you need to do is, if you have already taken a subscription, you can enroll. Okay, so if you haven't taken a subscription yet, you can subscribe and start taking these classes, start attending these classes, so that you're actually initiating your class 9 since it is the beginning. Okay, and introducing community. So never miss on any update from your favorite educator. So get session notes and practice material. Share your feedback directly with the educator via polls. Okay, so this can also be done. All right. So now Bugs Bounty. So this is opportunity for all learners to report any inappropriate content in the video. So be the first one to report a particular issue to claim your prize. Report any inappropriate content using the form in the description. All right. So now how you can watch my special classes. So all you need to do is download the Unacademy Learner app. Open the app and click on Get Started. Enter your mobile number and the OTP. Sign up using your email ID. Okay. So how you can watch my special class? So all you need to do is, again, after you follow that step, select the class 6 to 12. Then you can select the category ICSE. Okay, so then choose the language of your preference, like English, Hindi, English. So once you do that, so you can click on notify me on the special class. so that you will get notified about the session 10 minutes before. So you can also use my code VANIYT. So this is my code. Okay, so with this code you will be getting 10% discount on the subscription. All right, so now since we are done with today's seed structure, so any doubt you have, you can get in touch. So that is through the Telegram. Okay, so Telegram link I have added in the very beginning. So that is, you can just search in Telegram Vani Kari ICSE class 9 and 10. So there, I can send these notes. Okay, so click on bell icon to get notified about the classes. Subscribe to our channel, that is Unacademy ICSE and ISC both. So like and share the video and download the app now. So that is download the Unacademy Learners app, and this PDF anyway I can send it on my Telegram. Okay, so see you all in the next session. Bye bye everyone. Okay, so excuse my cold today. Alright, so see you all. Bye bye everyone. Take care. Thank you so much.